Hello, everyone, and welcome to Protein Biomarkers for Tumor Differentiation, Insights from Olink Explore HT. I'm Matt Thorne of LabRoots, and I'll be your host for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink. To learn more about our sponsor, please visit olink.com. We do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You can also submit your technical issues there as well if you're having any trouble seeing or hearing those presentations, and we'll reach out to you directly. I'd now like to welcome Ida Grunberg, Chief Scientific Officer of Olink.
Thank you, Matt. So very nice to be here, and I have the great pleasure of introducing Professor Ulf Gyllensten from Uppsala University, who have been a collaborator since the very beginning of Olink's history. And it's been really exciting to follow your research over the years, Ulf, with, I would say, pioneering publications on everything from impact of genetic and lifestyle effects on the proteome to prediction of biological aging based on lifestyle choices such as diet and exercise and smoking habits, to even forensic prediction on phenotypic traits using plasma proteins. And this obviously got some media attention. So that was fun as well. But of course, your main focus and drive has been in the field of gynecological cancer, and more specifically on ovarian cancer. And today, Ulf will be presenting their work on biomarker development and validation for gynecological cancer. After Ulf, I will share some more information about our Explore HT platform. But with that, I just want to say thank you for being here today, Ulf, and for all of you who are listening in. And the floor is yours, Ulf.
Thank you, Ida, and thank you, organizers, for inviting me to give this talk. Also, as Ida said, we've had a long history with using the Olink platform for various purposes. And this time, I will just concentrate on the gynecological cancer project, which is still ongoing. And we've been fortunate to essentially be early users of the PA technology and gone through essentially all the different platform versions and have some experience of that. So just initially, I'd like to just say that I have no conflict of interest, although we are collaborating with Olink. I have no financial links to them in any way. And I just skipped the first slide, so let me go back to that for a second. So this is actually a large collaboration between my own group and a number of clinicians in Uppsala and elsewhere in Sweden. A very translational research project in the sense that we're working with the clinicians in understanding what are the issues and what are the questions they would like to address, and how do we apply the technology that Olink has to address some of those questions. So gynecological cancer is obviously not one cancer, but actually a number of them, and these are the three main ones. Cervical cancer, which we know is caused by papillomavirus infections, ovarian cancer, and endometrial cancer, which have much more complex etiologies. We know their tumor mutations. We essentially know the site of origin, but we don't really know the causes in terms of the events leading up to cancer. Now, these three cancers, if you put them in one bag, essentially cause a lot of cancers in women. There are over 1 million women yearly or annually suffering from these cancers. And these are some numbers from the US from some years back, where you see the numbers in the US and the numbers in the world as a total. So 1 million is on a global perspective. And for the three cancers, about half of these cancers are cervical cancers due to HPV. And the other two are then divided up between the ovarian cancers, which about 250,000 women, and the rest is endometrial cancer. And the numbers on the right here tell you something about the survival rates once you get the diagnosis. And you can see in cervical cancers, we do have organized screening programs, which has alleviated the burden to some extent. We have a 5-year survival rate, which is approaching 90% in this data. It, of course, varies globally. In some cases, we have much lower, depending on the situation. But if you look at ovarian cancer, that stands out as the cancer with one of the lowest 5-year survival rates, less than 50%. So this is a very grim cancer, and the reason for the low 5-year survival rate is, of course, that we discover the cancer based on symptoms, and the symptoms behind those is usually a severe stage 3 or 4, and the likelihood of survival for long periods of time beyond when that diagnosis is set is very low. So if you look at the clinical side of these things, we do need different tools here. If you look at ovarian cancer, we would like to have a diagnostic tool, biomarker diagnostic tool, by which the clinician, when they make their call on this cancer, can decide whether this is a benign form, benign cysts, or various types of stages of ovarian cancer. So this is a cancer which is discovered mostly by diffuse symptoms, pelvic pains, and bleeding, and they have to go through ultrasound, transvaginal ultrasound, and based on the transvaginal ultrasound, the clinician makes a call of whether these are benign cysts or different kinds of stages of ovarian cancer. And in most cases, if there is what they call adnexal mass, some aberrant ovarian symptoms or views from the transvaginal ultrasound, they decide on surgery to determine whether this is a benign form or not. And so in Sweden, at least, about 75% of that surgery actually results in a benign diagnosis. And since the surgery is always sort of comes with dangers, risks, side effects on fertility and other things, there is a need for a biomarker test that would aid the clinician and maybe allowing them to avoid the surgery when there is a benign cyst instead. We have a sort of a similar situation for endometrial cancer, just to give that. It's discovered by symptoms, pain in the pelvic area, abnormal bleeding. In most cases, if it's postmenopausal bleeding, that may be a risk factor. And again, we have different kinds of outcomes here. It's either a severe form of endometriosis or it's a low risk or high risk form of endometrial cancer. And the clinician, if there was a biomarker test at that point, could use that to stratify the patients on different types of follow-up. So we've been involved mainly up to now with ovarian endometrial cancer in terms of biomarker discovery. And there are really 3 targets for these biomarker developments. One is to make the diagnosis more precise, as I just described, for symptoms, to distinguish the benign and the malignant forms, and to reduce the diagnostic surgery. At this point, there's no biomarker. We'd also like to allow for better monitoring of the relapse. As I said, the outcome once you have a treatment for for ovarian cancer is still pretty grim. The 5-year survival is less than 50%, and there are no biomarkers to follow up. So if we had biomarkers that allowed early detection of relapse, those could be used on a monthly basis and discover relapse earlier, and then hopefully extend the lifespan of those women. And then finally, we would like to have markers for population screening. This is the only way in the long term to reduce the incidence of this cancer. And of course, this is a tough call. We need to find the cancer at early stage before the development of symptoms. And right now, there is no biomarker which has sufficient sensitivity and specificity for this. CA-125, the only marker which has been tried, really lacks sufficient specificity to enable these types of programs. So when we started looking into this, we actually came in from the other end. We started with genomics. And as a background, my background is really in genomics, but we saw that there was a wave of proteomics coming around. This wave is based on the fact that there is these new technologies for high throughput screening. And you could see the parallel developments that we've seen in genomics, that instead of having to look at individual genes, genomics allowed us to look at the whole genome. In a similar way, instead of looking at single proteins, we could now use these high throughput proteomic screening technologies to look at many, many proteins at a time. We can also combine single proteins into multiplex protein risk scores in a similar way as the polygenic risk scores are being handled in the genomics field, and so forth. So we saw the parallels, and we decided to go for proteomics in addition to the genomics we had already worked on. And in terms of our study design, it really is a fairly simple and straightforward one. in each of these cases, we've started off with the discovery phase, trying to look at as many protein candidates as possible. Most of our work has been done with the Olink PA assay. We've also worked in mass spec to some degree. And then based on the top candidates coming out in the discovery, comparing cases and controls, or in our case then, the benign women with benign changes of the ovarian type, benign cysts versus the malignant ones, we do get a lot of candidates which have significant differences in levels. You take them through several independent cohorts to validate that they are actually not significant just due to statistical error, but true positives. And then we have converted the final biomarker sets into an absolute quantification score. So you can actually look at individual samples, combine data sets from different runs, and have the runs done in different laboratories to see that the values are coming out in the same way. And then we're now into the fourth stage with many of these biomarkers in terms of clinical validation and throughput. But essentially all the PA platforms that we have used have gone through the same set of steps. So some years back, before the HT came around in 2019, we had gone through the panels that were available then and identified an 11 protein panel. These are the proteins on your right-hand side there, that if you put them in together into a multiplex protein assay, they and use those to see whether you can distinguish the benign cysts from the malignant forms of cancer, ovarian cancer. They performed quite well. We had an AUC under the curve of 0.9. We had sensitivity and specificity values that were quite good. And they seemed at that point to be good candidates for the improving the diagnosis of women with the adnexal ovarian mass that I talked about. So if you look at that data a little closer, you can divide them up, and we calculated what we call a prediction score, which goes from 0 to 1 in terms of predicting cancer. So what you're seeing here is a graph where we have the women in the study color-coded depending on whether they had a benign cyst. The gray one are the borderline ones, the ones that the clinician could not decide on whether they're malignant or not, and the red ones are the ones increasing stage of severity up to the very dark red. You can see that if you had a score of about 0.5, you were on the green side, so to speak, whether it was above 0.5, 0.55, and upwards to 1, you were really on the malignant score. So this is the way we actually try to use the data. Instead of looking at individual proteins, we have a combined prediction score based on the multivariate analysis of these 11 proteins. This is another way of looking at the data. We have, instead of looking at them as risk scores, you look at the different stages of the cancers. So on your right-hand side, you can see we can easily identify women with stage 2, 3, and 4 from this blood sample. The only stage which is a bit in between here is the stage 1, the early ones. We could see that we can identify some of them, but it's not a clear-cut discrimination yet between the borderline benign and the more severe types. So using this 11 protein, we then went into a phase where we where we converted this into an absolute quantification assay, and again took a look at patients. And in this case, we also made use of the fact that some of the biobanks we're using have access to samples collected over time. So on your left-hand side, you can see the clinical history of individual women from the point when they were diagnosed, the black spot there. Each line is a woman's clinical history with the different stages that she went through up until, unfortunately, in many cases, within the follow-up time, they died. So we could take women and we can look at the different categories of management, from the primary tumor at the time of diagnosis to an ongoing treatment, to the responding ones, the final treatment, and then to those that relapsed. So on your right-hand side, you see the groups of women and their risk score at that stage. So initially, the black dots shows that they had, on average, a very high risk score at time of diagnosis. the risk score decreased substantially during treatment, and after the treatment, right after them, they come down, almost all of them, to a very low risk score. And then unfortunately, the risk score increased in connection with the relapse. So essentially, this told us that the risk score and the proteins that we are looking at are actually following the clinical course of the individual patients. And then we decided to say, can we look at patients not as a group, but actually identify different trajectories for different subsets of the patients. So this slide shows when we let a computer identify subsets of women that have similar clinical trajectories and what they look like. So in this case, the computer identified 4 different types of clusters. The first cluster, which was about 10% of the women, they had a very high risk score at the initiating point. You can see that in the black line there. They didn't respond to treatment and followed a very high risk score. And essentially, during during the period of follow-up, essentially all of them had died, unfortunately. So they were the non-responders, essentially, to the treatment. In clusters 2, you see different trajectory where they did respond quite quickly. Initially, the risk score dropped, but unfortunately, after a fairly short period of time, it increased again. So by the time we had this particular study follow-up period terminated, about half of these women responded. So these were really relapse women that didn't unfortunately survive very long. And then we have clusters 3 and 4, which had slightly similar trajectories. They started for clusters 3, very high risk score, then responded very well, and then stayed down, although there's a slightly slope upwards. So these are women, if you had a monitoring case where you can monitor them over time, you might be able to see that there's a slight relapse coming, and you could intervene with a second round of treatment and still keep them alive for quite some time. And for cluster 4, which is actually quite a few women, almost 35 or 36%, they started off with a relatively low or medium risk score. They went down, the treatment worked very well, but unfortunately they started to approach a higher risk score over time, although it took some time. So again, this is a group of women where you could intervene quite substantially. So this was one example where we were able to use the risk score to sort of evaluate what does it tell us about the clinical history. Now, given that the different platforms have come around, we've also used all the present platforms. And of course, with more proteins, there are more candidates. So this is just a graph showing you the number of candidates that have come out of different types of analysis. So the previous slides were actually based on a fairly small set of proteins, the initial panels that Olink made available. And then we tried the 1536 panel, the ones that have about 3,000, what they called Explore Expansion, and then lately Explore HT with 5,300 proteins. And the bars really tell you the number of candidates that have come out of these analyses that can be used to distinguish benign cysts from the ovarian cancer. And these are corrected p-values, or the number that have been corrected for the number of tests performed. And this shows you that, we had initially maybe 30 or 40, and when we turned to the 1500, we've gotten around 100 proteins that came out. We turned to the 3,000 protein assay that doubled, and so forth. So initially it looks a bit scary. How many protein candidates can you find? But it turns out this is really a very large benefit in the sense that the more candidates that you have to start evaluate, the more flexibility you will have in actually identifying the best candidates and the most appropriate candidates given the different circumstances that exist in the clinical setting. So based on this 5,300 analysis, the HT analysis, we know how many candidates, and we can prioritize them based on their stability over time, based on their dynamic range, based on their clinical sample type that we tend to use, and based on the detection pathology that might be useful. So we actually get a lot more flexibility in going from the primary data to a test that might be useful. So in terms of the HT analysis, we are still they have still not been published, but I added a reference to the Med archives where the first paper is actually deposited right now. So if you want to look at the data, you can do it there. So this is some of the early analysis from using the Explore HT. Again, we're looking at the difference between benign diagnosis and ovarian cancer. And the three graphs shows you on the left-hand side when we look at the benign versus early stages. There's a few proteins that comes up quite in the volcano plot there. In the middle one, we have the benign versus the late stage, and of course, there's a wealth of proteins. Many of them are sort of the same that we found before, but we've actually found quite a few new ones that needs to be evaluated, which looks really interesting. And then on the right-hand side there, we've added all, so it's benign versus any stage cancer. And again, we have many good candidates there. So one thing that we've also done is to take a look at when we have so many candidates, what are we actually seeing in the plasma proteome here? What is that reflecting what's going on in the tumor? So we've actually done a fair amount of RNA sequencing on the tumors from these patients, and I'd just like to share just a couple of points there. So this is one slide looking at the correlation between the plasma protein that we've just looked at, and then the abundance of the mRNA in the tumor from these specific individuals. And these are three examples where we find strong or not so strong correlation between the levels. Now, this is it's a bit hard to evaluate, but what do you expect? Well, obviously, if you want to use a biomarker, it's always gives you confidence when you have a strong correlation between the levels in the tumor and the levels in the plasma. But of course, some of these proteins are not acting directly. They're acting through interactions and in many steps of interaction, and we may not necessarily predict a strong correlation between the protein levels and the mRNA levels in the tumor. And again, we have much more data, so this is just a subset of the data. In some cases, we do not find a strong correlation between the two types of levels. It doesn't mean that the biomarker, the plasma protein biomarker, is a bad biomarker. It just means that it has to act through some other level of regulation. So this is one more study example from the HT. This is again the plots that we've done, the ROC curves. And then the one side, we've actually looked at the ROC and divided them up on the different histology levels, or histology types, I should say, to see whether we actually have a higher performance for a certain type of ovarian cancer histology. And it doesn't seem like that. It seems like it's performing reasonably similar between the different histologies. The main and the most dangerous one is the one which is in dark blue here. And on the left-hand or your right-hand side, we have it divided up on the different stages. And you can see that the performance is quite similar with the exception of stage 1, which is the yellow one. And this is, again, the most difficult one to detect. It's also the one, unfortunately, where we have the fewest samples. So I don't want to say that the biomarker assay we have is not performing well. It might also be a sample size issue at that point. So where are we in terms of our development of a diagnostic? So I just wanted to put this in the context of existing diagnostics. So this is on the top, you will see just a graph on the biomarker, multivariate biomarker model that will now come out of the HT. So the previous model and most of the data that we published have actually been on the 11 protein plex that I showed you initially. Now with the HT, we've actually been able to reduce the number of proteins, replacing a few proteins with a few other ones, and then come down to an assay which is only 8 proteins. And of course, the fewer proteins you have, the simpler it will be to synthesize and use. So in the top graph here, you see the distribution of the different cancer cases with the different stages relative to the risk score for this 8 protein assay. And on the left-hand side, most of them are gray, which means that they're benign. There are a few cases which are still in that risk score level, so it's not a perfect distinguishing biomarker test yet. We're still working on trying to refine it a bit, but it looks quite good. And if you compare what's available in the literature and on the market, that's shown in the table below there. So there are different tests that have been developed, and some of them are commercialized. The ROMA is really not a test, it's an index where you use some of the existing biomarkers along with some clinical information, and the sensitivity and specificity are given there. Overa is a test which is based on, as you can tell from the bottom there, 5 proteins. And it performs reasonably well in terms of specificity, but a fairly poor sensitivity. And the Gallery test is really the one which is in the Grail, which is manufactured and provided by Grail. It's hard to get a sensitivity there, but their specificity, this is a multi-cancer test, I should say, so it's not specifically designed for ovarian cancer. But at least in this context, it seems that our test, or our diagnostic that we have produced so far, performs reasonably well, both in sensitivity and specificity. So we're actually quite happy with that, and now taking this on into new stages. And so what are we going to do with that? Well, as I said, it was initially developed to diagnose women with benign and malignant changes upon the transvaginal ultrasound. So now we're clinically going to validate this new protein assay in the similar way that we did with the 11 protein. We also started a project to monitor for relapse. So this is a study where women that have gone through therapy and are at risk for relapse will be given a paper card and a little lancet to take a blood sample, finger blood sample every month, and send it back to the lab to see whether we can detect signs of relapse earlier than the present stage follow-up strategies. So this is ongoing, and we're quite excited about this opportunity. And then, of course, we would like to see whether our assay has potential to be used in population screening. And to do that, we need prospective cohorts. So we're both exploring existing prospective cohorts to evaluate the assay, and we're actually also establishing our own prospective cohort based on DBS cards and blood sampling to be used in a year or two to evaluate the ongoing developed biomarkers for ovarian cancer and endometrial cancer, I should say. So with that, I'd like to just end with a picture of the team here. As you know, developing these things is not a one-man show. This really takes a village to develop it. And so this is my village, a lot of collaborations and collaborators on the clinical side, along with people on the preclinical side and on the side of developing statistical methods. So with that, I'd like to close, and thank you for your attention.
Thank you, Ulf. That was a fantastic presentation with very encouraging results. So thank you for all of the great work that you shared with us today. So now I want to continue to speak a little bit more about our Explore HD platform. So I've been with Olink for more than 13 years, and it's been an amazing journey. And currently I'm leading the HiPlex portfolio initiatives, including our flagship Explore HD. But today I thought I could just start by very quickly summarizing the primary reasons why scientists are using Olink. So in short, we want to reveal new biology and develop better therapies with a proteomic platform that delivers and that the community can trust. And what do I mean with that our platform delivers? So this is a nice slide. So in just a decade, there are more than 2,300 publications based on our proximity extension assay platform. And this number is rapidly growing, covering all disease and application areas. And this is something we are extremely proud of, as it ultimately serves as proof that the platform delivers and helps drive precision medicine forward. And some encouraging statistic is that we also see a strong increase in the impact factors of these publications. So not only are there more and more papers, but also with higher and higher quality. And one example of a project using our platform with an enormous impact in our community is the UK Biobank Pharma Proteomic Projects that conducted one of the largest proteomic studies to date with more than 54,000 UK biobank samples, revealing biological insights into health and disease. And this impressive initiative generated data leading to 3 papers in the same issue of Nature last year. So first, constructing a genetic atlas of the plasma proteome. Secondly, also uncovering more than 5,400 rare pQTLs, important for drug discovery, enabling associations between rare variants and disease. Thirdly, also published data from their large-scale proteomic platform comparison. And finally, the consortium provided the whole scientific community with an open access population-scale proteomic resource. And the data truly enables fast publications, because we already see more than 100 publications just during the first year. And what all of these papers have in common is that they are based on our PA platform and our products. So I just want to go back a little bit and talk about proteomics. So it's a very complex field, and the proteomics field was suffering from technological challenges for many decades, and Ulf was leading to that as well, and it was sort of lagging behind genomics and transcriptomics when it came to coverage and performance, throughput, etc. But it's those fundamental challenges that we have worked to address and that our platform is solving for. And here I will just try to summarize them at the highest level. So first, when detecting proteins in a sample, proximity extension assay is requiring dual antibody recognition together with DNA barcoding to give rise to a signal. And this really drives the exceptional specificity of our platform with no cross-reactivity. On the sensitivity side, it's in general similar to ELISA, but we have some critical assays, some of the interleukins, for example, where our performance is on par with clinical assays, where the higher sensitivity is needed for some research questions. Dynamic range of the library covers 10 logs, so from femtogram to milligram levels of proteins. And we have a strict validation process of each target in our library, and I will go through it a little bit more on the next slide. For our Explore platform, we are using NGS as a readout, which enables high throughput, meaning that we can do tens and thousands of samples in weeks rather than months. And we can also piggyback on the cost evolution in NGS to lower the sequencing cost. We use minimal sample volumes of precious samples, and basically any matrix can be used and have been run. And finally, we are scalable. So in other words, that we use the same core technology from discovery to clinical translation. We take great pride in our performance and our ability to measure what we say we are measuring. So we want you to trust the data. So all PA assays undergo a 3-step 15-factor analytical verification process, where we assess all performance criteria such as sensitivity, specificity, precision, scalability, etc. So out of the more than 15,000 proteins assays tested, 5.4 thousand have been approved and included in products, whereas the remaining are still in the pipeline for future products once they have reached our high quality standards. So I would say that Olink is quite unique in a way that we provide the same PA platform from discovery to clinical translation, and this scalability comes with many advantages. So we have our HiPlex Explore products. We have the target portfolio, which plays in the mid-plex proteomic space, and finally our custom offering with our Flex and Focus products that allow you to design your own panel based on your research question. And we heard one example from Ulf that you so nicely presented. So it's one platform, but really endless of opportunities depending on your need. But let's go into more details now about our HT platform. We launched it roughly a year ago. But to start off, we increased the library of assays, reaching a total of 5,400 proteins. So we introduced roughly 2,500 new proteins. And we took a big leap forward for all the labs out there running Olink with a much simplified workflow also allowing for 4 times faster process of samples compared to our previous generation of Explore. And the throughput is also around 4 times larger, so we can run roughly 2,000 samples per week with 1 sample prep line. And finally, with Explore HT, we also did major improvements on the QC side with our Explore software. So now it's highly automated, robust, and provide actionable data in minutes compared to previously hours. Just to put these numbers in perspective, we can complete a study with 50,000 samples in just some months, generating millions of data points per week. So I think this is really fascinating when you think of the speed and the impact of data generated today from all the extraordinary biobanks and cohorts collected around the world. So we are very excited to take part of this and contribute to this proteomics revolution. So I already mentioned that validation of our assays before going into a product is of great importance for us. So when developing a new product, we want to assure that the high quality remains at the same time as we are adding more value from the new assays. So one way of assessing that quality remains over product generations is correlation testing using same samples across different products. So in this case, when comparing previous Explore 3072 with the HT, we see excellent concordance between product with a mean of 0.9 and median of 0.97. And another way of assuring quality is to compare precision across products using same samples. So here we were comparing intra and inter CVs for Explore 3072 with the same overlapping assays in the new HT configuration. So those are the two first columns that you see here. And as you hopefully can see, CVs are quite similar. And thereafter, we compared it with the new assays added into HT, and finally with the complete HT configuration. And as you can see, once again, with the same close range of CVs. So we conclude that HT delivers on precision equally well as the previous Explore product. And I should say that concordance and precision are just 2 examples of what we are testing. So if anyone is interested in seeing more comparison data between products, just reach out and we would be happy to follow up with additional data. But going into the value of the new library, here you see some examples of increased protein content added to our HT platform. So we have always had a very strong focus on inflammatory proteins, but we still managed to increase this with 7%, which we're excited about. And we also see a need to include more tissue-enriched markers. And as a result, we have increased this segment with 40%. And finally, we have also done improvements on our drug target content. So Explore HT includes many new therapeutics, as well as targets currently being evaluated in clinical trials. So to summarize this slide, HT offers the same high quality as previous generation of Explore and contains 2,500 new protein assays, delivering greater value for our community. So if we then zoom out and look at the complete HT library, it covers all major biological pathways. So we are using the Reactome database for better understanding of our current coverage, as well as where we have gaps that we need to focus more on for future products. So this is a useful resource for us at Olink, but also for our community. So we are also offering this as a pathway browser at our digital platform, Olink Insight, for everyone to freely access. So there you can search for specific proteins, pathways, products, and connect all information into a comprehensive summary. So this is a really great tool that I recommend everyone to take a look at who wants to reach more insights faster. So again, with HT, if we break it down, here you can see more numbers of protein assays across key biomarker categories, again with a strong focus on inflammatory proteins, secreted and tissue-enriched proteins, and drug targets, and also, of course, more in general, disease-associated proteins. And for this type of annotations that we do, we are relying on established databases to assure that we are focusing on the most relevant targets in our products. So how does Explore-HT reflect the human proteome? Well, the human subcellular proteome has been mapped by, I would say, one of the most respected references, so the Human Protein Atlas. And when comparing their map to our Explore library, we see that they mirror each other quite nicely with regards to the split between intracellular proteins, membrane proteins, and secreted proteins. And this gives us great confidence that HT provides a good representation of biology. So we see it as important to include intracellular proteins, since they do represent 2/3 of our proteome, but they are found inside the cells. And because of that, they are not expected to be readily detected in plasma. However, of course, they can occasionally end up in circulation, maybe in a setting of tissue or cell damage or rare conditions. And it's in those situations where it's critical to measure them correctly. And we see beautiful examples of that from studies conducted by our users. But I think this is an important topic, as some might see it as a concern, not detecting all proteins in samples. But then I think it's important to remind us on biology, and intracellular proteins are present inside intact cells where they serve their functions, and they should not be detected in healthy plasma. So if such proteins would be detected in healthy individuals, I would be concerned about false positives. So here we can also, of course, provide additional support by sharing performance data from our assay development, as well as insights from running other matrices. So, for example, cell lysets, where we more realistically can expect signals from basically all intracellular proteins. So all of this together gives us confidence that our platform will detect those important biomarkers if leaked into circulation to signal early disease risk, organ failure, drug response, etc. So leading to that as well, so detectability is heavily impacted by sample type and biology. And to illustrate this, here's a Venn diagram showing overlaps in assay detectability for plasma, cell lyset, and sputum samples, all analyzed with our Explore-HT. So despite the small sample size here that you can see, we detected 74% of all proteins in at least one of the matrices. And of course, powering your study will increase the chance of detecting more proteins and revealing true biological signals. So finally, I just want to share some more data from a study run using HT. And this was done in collaboration with the Broad Institute and their DAPMap project, with proteomic mapping of more than 160 different cell lines, representing the diversity of human cancers from more than 20 different tissue. So the first thing we did was to evaluate correlation to our previous Explore 3072. And we're happy to see a median correlation coefficient of 0.96. Thereafter, we did pathway analysis to assess if the cellular profiles reflected the tissue-specific pathways. And once again, we were excited to see enriched pathways representing the biology of the tissue of origin. And the colors are not great on this slide, I see, but we maybe update that to the on demand. But if you look to the right, I think it's really fascinating if you see in the lower end of the figure, it's the tissue origins with the enriched pathways labeled and highlighted in different colors. So if you can't see it, I can take an example. So brain tissue are supposed to be shown in yellow, and they had enrichment in the neuronal system pathways. And another example is lymphoid that are marked in blue were enriched for immune system pathways. And with the same data set, we also wanted to go back to the question about what biological value the new assays introduced into HT provides for separating different tissues using UMAPs. So here, quickly, we first analyzed the set of assays overlapping with our previous Explore. So it's around 2,800 proteins, and could see good separation of the different sample groups. As you can see from the differently colored clusters representing different tissue. And when we did the same analysis, but this time only with the newly added HT assays, around 2,500, we were positively surprised to see how well these proteins by themselves could tease out the biological differences between the sample groups. And then finally, when we repeated the same analysis with the full Explore HT library, we noticed superior separation of the different tissues, and concluded then that HT provides the best biological insights. So this is my final slide. So if you're new to our platform and are wondering how to get access to it, you can reach out to us for more information about our analysis services. You can go to olink.com and find a service provider in your region, or then you can join the community and set it up in your own lab. There are more than 300 labs around the world running and providing our platform, and we are happy to support each and every one of them. So with that, I just want to say thank you for your attendance, and it's time for a Q&A. And I think we will have my colleague Agnieszka joining for this session as well. Great, here you are, Agnieszka.
Yes, thank you so much. It's my pleasure really to join you in the Q&A sessions. We've received many questions, but we have time to receive even more. I see different topics appearing in the questions, so we'll group them and go about those. Maybe we'll start with some quick one. We've been asked if we have a technology access program. Ida, you already answered this basically before, but do you want to repeat a second for anybody new? If they would like to start, what do they do?
I should say I should contact my colleagues. I'm not on top of the process behind it, but we do have a very good program for it. So reach out and someone else with more expertise in this can guide you. But yes, we have it, and it's been very appreciated.
Okay, very good. And then, so there are questions related to the Explore HT library, where to find it. We can certainly send out the link. It's again on the webpage. And Ulf, I'm coming over to you. There are questions related to the signature. Could you tell us more about the proteins that you identified as final, the 8 final? Are they plasma proteins or are tissue specific, intracellular of origin? What do we know about those?
Thank you for the question. Yes, it's a good question. I mean, it is a mixed bag. I mean, one of the things we have realized, and ovarian cancer may be specific in that sense, as we saw, there are many different candidates. So one of the tasks initially is, of course, how do you pick the best ones? And the best ones are not necessarily the ones that all give the strongest p-value, because they together have to identify the cancers. It's not like we use them one at a time. So we're using a multivariate and sort of machine learning algorithm just for the selection of proteins to identify the best set of them. And that's not really the answer to your question, but I'm getting there. So instead of going through all of these hundreds and hundreds of candidates that we now have with the HT and trying to identify their origin and their purpose in life, we're actually going through the computer selection process first to identify the best set, because that becomes a more manageable number. Initially, it's more than 8 because we want to reduce that eventually. And then we bring all these other criteria in, as I said there, with the different types of dynamic range and quality and stuff like that. And then at the end of the point, when we have identified them without actually looking at the proteins, what are they doing, we take a look at what's known about their sort of location. Are they in plasma only? Are they intercellular? And of course, the majority of ours are actually plasma proteins, as far as we can tell. But it's sometimes some of them have been described in the context of ovarian cancer. So some of the proteins that have actually stayed at the top list all the way from the early PAA platforms are still there. They're still good candidates. They're known to be elevated in ovarian cancer since before, both in terms of some of the data I showed on the PAA level and in the plasma, but also in terms of their mRNA regulation. So we're pretty safe in saying that these are good candidates. But then on top of that, we have a number of them which are not easily identified from the literature as being involved in this particular cancer or cancerogenic process. And I think that's, if you have many candidates, that's going to be the case, because again, we know very little sometimes about the interaction of proteins. So one of the things I didn't have time to show you was really this interaction map. You take all these proteins that show, positive signals in Bonferroni corrected. We still have 500 proteins or a few hundred proteins there. And you look at the interaction, and if you look at the interaction maps, you see that they're actually correlated. Some of them correlated in groups. And it may be that the origin of this whole sort of process that we're looking at is down the line somewhere, but it's not the one we've picked necessarily, because it doesn't really matter which one you pick if the levels are correlated and if there are interactions through this process. So that's a long answer, but I think what we've learned is that our understanding of the plasma protein is still pretty limited. And if you start from the other way around, you start looking at the proteins and you try to identify which, from a functional point of view, seems to be the most interesting. The only thing you do is rely on the present literature, and that is, in terms of our proteome, quite limited. Even there's a lot of literature, but it's quite limited in terms of effect of each individual protein. So we benefited from the fact that there are many candidates to pick from, and hopefully the ones that we're eventually going to be using now in the future are plasma proteins, or at least excreted in plasma at one point or the other. Going back to what Ida was saying, I mean, of course, we would like to be able to measure them in a substantial number of individuals, not necessarily the ones that are very rare and just occur in one patient or two patients. We'd like them to be a signature of a patient, but the combination of protein is the readout. It's not a single protein. So it may be that certain subset of cancers, or ovarian cancers, have specific signatures, and by combining them, because we're looking at the whole set of patients, we're actually trying to get the best out of this enormous range of proteins that are available for selection.
Absolutely.
Now I know in many cancers there are not that many proteins to select from, because there are not that many proteins that show up in these kinds of studies. But at least for ovarian cancer, it really is a very heterogeneous picture.
Ulf, I just had a follow-up question to that as well. So it sounds like there is a combination of known markers for you, as well as some novel ones. But like the CA125, is that part of the 8 protein signature?
It is. I mean, we've really struggled with that protein, partly because it's the only one which is a well-known candidate for ovarian cancer. So we wanted to have it, if possible, if it showed up, we wanted to bring it in. And this is partly because we're, the clinicians, when they get to the point of using a diagnostic, they would like to know what's in it and what's going on here. And if they feel that we haven't even used their sort of classical markers, they become a little more resistant for good reasons. But again, I mean, we could have picked other combinations which have performed equally well as having the one with CA125 in it. Now, CA125, as you know, is a difficult protein to measure. So from a protein detection point of view, it's not an ideal marker, but we have it in there, and we need to bring it in, I think, to convince the clinical collaborators that this is a reasonably accurate assay.
Good, good. Very interesting. There is a question related again to the biomarker signature, the group of proteins you identify. Do they indicate any pathway that may be involved in cancer progression? And I guess this is also connected to what you were saying in relation to using now this signature to identify, early detection for relapses or, for in future. So can you comment on this, please?
Yeah, I mean, we have a couple of proteins which, if you just take the data and essentially instead of having all the stages in there, you just take stage 1 versus the benign cases, as in a discovery study, really. Then they show up. There are a couple of them that show up associated, not extremely strongly associated, but they're in there. So we would try to, in the overall sort of balancing of different proteins that should be, we will try to bring them in because, again, as you say, those would probably potentially be quite valuable for the early detection in a screening setting. It's an interesting question because in most of these studies, if you were to design a study specifically for screening, you might think that the best way to do that would be to have only normal versus the early stage cancers. Because that would, you would seed out the best candidates for early detection that way. That's not the way most studies are done. Most studies in the literature have full-blown cancers as one group and healthy people on the other side. And of course, with that setting, you're very likely to find markers, but it's not clear that those markers might be the crucial ones for early stage detection. So having a large cohort of patients and having detailed information on the different stages or different histologies or whatever is in there is crucial to make this into a good biomarker discovery project.
Thank you. Thank you. I want to take the conversation around something that Ida mentioned before about being concerned with regard to potential false positive if you don't have the specificity. What would be the impact, Ulf, if there was a technology that was not so reliable in terms of specificity?
Well, it depends on where you use it, but of course, if anything that has to go into the clinic, we are working on versus the clinic, but it's clearly not an assay which is being used full scale within the clinic, and they put a lot of pressure on us but sort of coming from the preclinical part that the assay has to be foolproof and foolproof technically speaking too, both in terms of the technical sensitivity and specificity and on the clinical sensitivity and specificity. Now, it depends, of course, in which setting it's coming in, but if it's going to be something that guides the clinician in terms of treatment or non-treatment or stratification, it has to have very, very high performance levels. And again, to the point that Ida was making, we have looked through our data on these different platform levels, PA platforms, to see, did we find, what was the performance? Was the correlation between the levels that some of these proteins had with the cancer in the early PA levels, in the 3000 expansion, in the all these levels, and the correlation has been really, really good. So it shows that even though you guys have been able to increase the multiplexing in a very amazing way, the performance of the individual proteins that we have identified and followed along have performed in a very similar way, and that's comforting because you always worry when the multiplexing goes up as it has that you would lose quality on the individual markers, but it doesn't look like that has happened, which is very comforting.
Thank you. That's very reassuring, for sure. We have a quick question, Ida, related to what biological fluid are compatible with the Explore HT platform.
So we have a good experience across many alternative matrices with both the previous 3072 and HT. So we run, I presented urine, sputum, and plasma serum, of course, dry blood spots or different filter applications. Ulf also brought this up, lysets. You name it. So our general guideline is that everything that can be solubilized can be run, maybe with the exception of fecal samples and formalin-fixed tissues. So it's really flexible in that way. And also since we're using such tiny amounts, we can also provide very comprehensive analyses on these precious sample types within pediatrics or microbiopsies or tape strips in dermatology. So it's really wide spectra of matrices that we can run.
Wonderful. I think we are out of time. Before I pass over the word to our colleague at Lab Roots, I just want to share a couple of comments here. Many people are saying thank you for the very informative and excellent presentations. And also someone is mentioning that the sensitivity and specificity of this assay is already meeting the FDI requirement for CDX assay, just based on the data. But obviously our product, our research use only so far. And over to our colleague. Thank you very much, Ida and Ulf. There are other questions, but we'll come back via email with the people who made them. Thank you.
Thanks for having me.
Yeah, I'd like to thank our speakers for their time today and their important research. We'd like to thank Lab Roots and our sponsor, Olink, for underwriting today's webcast. Before we go, we want to thank the audience for joining us today and their questions. Any questions that they didn't have time for today or that are submitted during the on-demand period, those will be addressed by the speaker via the contact information you provided when you registered. This webcast, like I said, will be able to be viewed on demand. Lab Roots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who missed today's live event. Until next time, thank you very much and goodbye.